Hey guys, alright, part two of the review. Sorry, of the review. Um, just a couple more things on base pair. Sorry. Just a couple more things on base pairing. So, she might ask something about like how many hydrogen bonds are in between each set of nitrogenous bases. When she talks about nitrogenous bases, this would be your A and T and your C and G. So the way I remembered this one is A and T both have two lines. I mean, this one's got that one, but like, whatever. So that reminded me that they have two hydrogen bonds between them. And then on this one is, I put like C on top of G, which would give you three. Kind of weird, but that's my best way of remembering it. So that's how I would remember it for that one. Okay, next part. We're going to talk about protein synthesis and ATP and nucleic acids. So this would be your ATP, right? Because it's got 3 phosphates. This would be your nucleic acid. During my, when I took the class, she did ask a question really similar to this, and I kicked myself in the foot for like forgetting this. But if she asks like what the structure of ATP is most like, put nucleotide. Because as you can see, they have their nitrogenous base, and they have a sugar molecule, and they've got phosphate on it. So they're a lot like ATP is a lot like a nucleotide, except it's got 3 phosphates instead of 1. So the big thing that I didn't go over from chapter 2 yet is protein synthesis. She might not have gone like super in depth on it, but it's good to know. So she talked about like transcription and translation. The difference between those is pretty big. So with transcription, that's when you're taking DNA and putting it into RNA, so like transcribe, and then translation, you're taking RNA and turning it into a protein. And that's through dehydration synthesis. But I think she goes over that later on in the semester. I'm pretty sure. If not, we'll go over it right now. Let me double check my notes. Okay, we're just going to go over it now. If she didn't go over it into this much of depth, I'm sorry, but she will at some point. So, you have your DNA, right? So you have your DNA right here, and it'll be transcribed into RNA. And from there, it'll be translated into a protein, right? So it's going to code for all of the section of DNA that it should. And a good thing to remember is the start codon. If she asks you what AUG is, start codon. AUG. Always a start codon. So right here is going to be your mRNA. There's 3 different types of RNA. I really think she goes over this later. I'll go through her notes and the slides again. If I see a lot of stuff on this, then I'll send another link. Probably not one that I made. You guys probably are going to be sick of me by the end of this. But just understand, DNA is transcribed into RNA, where you would have AU instead of AT, which will then be translated into a protein to be used for multiple things throughout the entire, really the whole body, but for multiple processes. So, Chapter 3. I'm going to level with you guys. Chapter 3, I can't do much. I can help teach stains, I can help teach basic math, I can help with total magnification and the FA technique. That stuff I can help with. Specific microscopes, you're going to have to learn yourself, which again is another thing that you could use flashcards for. So, because I can sit here and tell you what a compound light microscope does, what scanning microscopy is, I can tell you what, like, electron microscopy is, I can tell you all that, but that's not something I can make you memorize. So, that one is on you guys. If you need help making flashcards, let me know. But flashcards for me, I would honestly do the important people of microbiology, and I would do microscopy. I'm sorry. I can't help more with microscopy. That's just a memorizing thing. However, I can teach you guys like total magnification. I can teach you how to convert from meters to millimeters and so forth, and I can teach you all the different staining, because that one I feel like staining is a little hard. That's another one that you could make flashcards for, but I'm going to give you a couple things to help understand why they stain that way. So, let's start with converting meters to millimeters and so forth. So, meters is going to be your basic, like, SI unit, right? We've heard about that in general chemistry. If you've taken general chemistry. If you haven't, I'm sorry. But, so, your meter is going to be your base unit. Millimeter, nanometer, micrometer, those are all smaller, like, parts of it. And the way to remember this is they go down by, like, -3. So, millimeter is going to be 10 to the negative 3. Micrometer will be 10 to the negative 6. And nanometer will be 10 to the negative 9. So, if she's asking you to convert from, like, millimeters to nanometers, just understand there's a difference of -6, like 10 to the negative 6 between those. And if you have specific questions, let me know. I can try to record like a solution video. But those ones are pretty easy, and I feel like most people understand that, especially if you've ever done like dimension analysis or just multiplying in general. But just understand they'll go down by about 3. And I think those are the 4 specific ones she mentioned, because those are really what we use in microscopy. Like, those measurements are, I think, the lowest that we use in microscopy. If there's one smaller, let me know. But I think the next one would be like 10 to the negative 10th or 10 to the negative 11th. I've only memorized those first 4. Okay, next thing. Total magnification. So, total magnification is the total magnification through the microscope. So it's your ocular lens multiplied by your objective lens. Your ocular lens is going to be like the part of the microscope that you actually look down into, so like where your eyes go, right? And the objective lens is the part that you can change towards the stage of the microscope, and that one is like the 4 times, 10 times, like oil immersion, all of that. So if she asks you like what's the total magnification, or she asks you what, like, she gives you like the ocular lens like percent, or ocular lens like whatever it is times, and then like says oil immersion, know what the oil immersion like amount is. So if she says your total your total magnification is, and I'll write this one down because then it's easier to see. So let's say she says your total magnification is 1200 times. Now, with most microscopes, your ocular lens is going to be 10 times, but you can't always assume that. So, let's say she says that your ocular lens is 30 times, and then she asks you for your objective lens. Super simple here. Take 1200 divided by 30. That means your objective lens would be 40 times. Does that make sense? It's super basic math. It's just understanding that your ocular lens and your objective lens is multiplied to give you your total magnification. There's that. Like I said, it's super simple once you get to understand it. And if you still don't understand it, like, I'm serious, feel free to message me. I know that the test is super, super, like, coming up super, super soon, and most of you are super worried about it. But she's not the kind of teacher that will go out of her way to trick you. She wants to make sure that you understand everything, which is why I said don't always assume the ocular lens is 10 times. But, like, she's pretty straightforward. I have really enjoyed all of her classes that I take because she is really straightforward and she's not going out of her way to trick you. Most teachers will go out of their way to trick you, I feel like. But she just wants to make sure you understand. So if you can pull from, like, sometimes she will ask questions that pull from, like, multiple topics that all relate. If you can understand how those topics relate and make those connections, which I'm trying to help with, then you'll totally be fine. Like, this test, I really like microbiology. That's why I've been tutoring this class for 2 years, maybe more. And that's why my undergrad research is microbiology, because it's really, really interesting. You just have to invest the time in it. There's my little speech. I'll get off my soapbox. Okay. Next thing, I want to talk about morphology. So when she talks about simple stains, basically when you use a simple stain, it's only to look at the morphology, so like how the cells are shaped. And you can use any stain for that, whether it's called carbofuchsin, emerald green, safranin, methylene blue, like, because you're only looking at the shape of the bacteria. We get into like more complex stains that help determine what bacteria there is, and I actually might link some of my specific gram staining that I've done to show you guys the difference between your gram positive and your gram negative, because it's super, super cool to see that. But with your morphology, what she wants to get at with a simple stain is just making sure you know what the shape is. So we talk about, like, there's 3 different shapes, right? You've got your circles, you've got your rods, and you've got, like, your little spirals. So your circles are called coccus. Way to remember that. Circle and coccus start with C's. And I'm pretty sure there is, like, if you look at, like, Latin or, like, root words, you'll probably be able to figure out why that is. I just personally didn't learn it. Then you've got your rods, which are bacilli or bacillus. Bacillus just depends on what, like, if you're talking about more than one or one, right? And then you've got these, your little spirals, which are spirillum. That one's super simple, right? Spiral, spirillum. Cool. So that's the basic morphology of it, like just the shape. But then you want to talk about the arrangement of it. So if you're talking about the arrangement of it, you've got clusters and you've got, like, stripes, essentially, right? So if you have, where is my brain at right now? You've got staph and strep, right? So with staph, you're going to have, like, clusters. And with strep, you're going to have chains. And the way I remembered the difference between those is when I was little, I got strep throat. And when my mom told me, I thought she said that I had stripe throat. So I thought that I had, like, tiger stripes on the inside of my throat, right? So strep, chain, stripe, same way, right? So that's how I remember that. But just remember that a cluster would be staph and a chain would be strep. And you can see that on, like, basic staining, which is super cool, honestly, if you've ever seen it. I think you guys will see it in lab if you haven't already. It's super, super cool to see, like, the way that they link together and to see the actual shapes, because it kind of helps put, like, visual to what you're learning. And I learned super well with visuals. I learned super well with visuals, so I think that's awesome to finally be able to see it, which is why I kind of want to show you guys some of mine that I've been looking at in my lab, because it'll help you to see a bit more of that, to have a bit more of a visual with it. So, we went over simple staining. We went a little bit over protein synthesis. If I need to go over it more in depth, I'll get you another video. And then we went over, let's see, we've gone over biofilms, people in microbiology, recombinant DNA technology, taxonomy, chemistry, bonds, pH, anabolic versus catabolic processes, macromolecules, protein synthesis, ATP, basic units, total magnification, simple stains, and morphology. So, this is where we start getting into a little more complicated stuff when it comes to staining. But staining is honestly really, really cool. Like, if you guys haven't done it in lab, probably will. But it's super cool. And I can give you a bit more, like, reasons as to why they work. So basically right off the bat, we're going to start with easier ones because when we get into acid, fast, and gram staining, there's a bit more steps in them, which is why they're a little harder to learn. But if you can understand why they work that way, you'll be golden. So with with negative staining, if you look at a film negative, everything that should like be light is dark, and everything that should be dark is light. Same with negative staining. What you end up doing is you stain the background, and then the cells remain unstained. So you can look at the cell and then not have to look at the background. So it's kind of like dark field microscopy in a way where you would have like the same kind of picture. You would end up seeing a dark background but a lightened specimen. However, this is a stain. And then we've got endospore staining. So endospores are super, super cool. What it is it's only with Clostridium and Bacillus, I believe. Clostridium for sure. But they, an endospore is kind of like a seed where when the conditions aren't right, it just shoves like all of its important everything, so its DNA, into a tiny little corner, forms a coat around itself, and then just chills. Like it'll lie dormant. When it becomes active and it's in an area where it can finally thrive again, it will only make one cell. I think the trickiest question she's ever asked was how many cells come from an endospore. One. I cannot stress that enough. One cell comes from one endospore. It's not like reproduction. It's not binary fission. It's not anything like that. It is one cell comes from one endospore. So when you stain that, what you end up doing is you will stain initially with emerald green, which will stain the endospore, and then you counterstain with like safranin, because safranin will turn red. Yeah. So you'll counterstain with safranin. So all of your living cells, right, will be red, but your endospores will stain green. So if you ever have a bacterial slide that shows both red and green bacteria, just know that you're looking at an endospore stain. I think that's it for those two. I know I went through them super fast. Just to recap, negative staining, you're staining the background and not the cell. And endospore staining, you're staining the endospores green and the regular cells red. Yes. Okay. So, next thing, let's do the FA technique. So the FA technique is super, super cool. It's used in fluorescent microscopy, and that's why fluorescent microscopy works. So basically what you end up doing is you have like antibodies, and we've all heard about antibodies, and I think they're super cool, and we'll learn a lot more about them later on in the semester with the immune system when we get into that. But you take an antibody, right? And they're kind of shaped like that. And what you do is you add like a fluorescent compound to it. So you've got like these little fluorescent compounds in here, and they will essentially bind to this, right? And then this antibody will bind to a bacteria. So then that is really not a good color. I need to not use that color. Maybe blue. Yeah, that's better. Okay. So you'll take these like fluorescent molecules and they'll be on a antibody, and that antibody will bind to a bacteria. So then when you start like going through the fluorescent microscope, like when you use this technique, when it like shines on it, these bacteria will appear to be glowing. And it's really cool. Like you can see it, I think the picture she shows is with Spirillum, where they'll bind to multiple places on it, so that when you look at it under the microscope, it literally glows, and it makes it a lot easier to see the microscope, which is super, super cool. That's literally it for the FA technique. Like you're taking a molecule that's fluorescent, binding it to an antibody, the antibody binds to the bacteria, and then you have your specimen that you can look at underneath the microscope a lot better. Next thing, acid-fast staining. So, with acid-fast staining, that's going to be used for mycobacteria. Because she talked about mycobacteria in the past and how it's got like mycolic acid within the cell wall, which is why this one works, is because of that mycolic acid. So, with mycobacteria, you're going to initially like put it in a little bit of a saline solution, and then what you'll do is you're going to put carbofluoxin on it, and then you're going to decolorize it using alcohol or hydrochloric acid, and then after that you're going to stain it with methylene blue. So your acid-fast bacteria are going to stay the same color as the carbofluoxin, which is usually like a pink or a purple. And then you're going to use your methylene blue, and that methylene blue is going to stain everything else. So the important thing, like in discerning between those, you're going to have blue for your non-acid-fast and pink for your acid-fast bacteria. And it's just a differential staining technique to help you, like if you have a culture and you don't know what's in it, but you suspect that it might like have an acid-containing, I guess that's the best way to describe it, an acid-containing bacteria, like Mycobacterium. I think we only use it on Mycobacterium. So if you think you have Mycobacterium, you would use that to kind of figure out if it's in there. Because if it's not, then it's not going to stain that color. And the last staining technique, which is my favorite, because I think it's so cool when you finally get like really clear results, is your gram staining. So gram staining is used to like determine if a cell is gram positive or gram negative. And that can be kind of confusing because I feel like gram positive and gram negative, like it just sounds like a bunch of words. But if you can look at a picture and like be able to understand what you're seeing in that picture, it'll help. I remember I think the first test I took, I missed a question a lot like this just because I didn't understand what the difference between gram positive and gram negative was. If I was like, I understood that like, oh well, one has more of a cell wall than the other one, but like looking at it in a picture was really hard for me to understand. So I have a better way for you guys to understand this because I did not understand it as well. So gram positive, think more, right? And gram negative, think less. Positive versus negative, right? So, gram positive cells. You're going to have this inner membrane in both your gram positive and your gram negative cells. And that's super important to remember. Just remember you've got that inner membrane. Then, with your gram positive, they're going to have an extremely thick cell wall. Like, really thick. Okay? Whereas your gram negative is going to have a really small, like, thin cell wall. That makes sense? That was a really bad drawing. Okay, there. So this is your gram positive, and this is your gram negative. So already you can see some differences, just between, like, how big the cell wall is. Right? So gram positive, inner membrane, super thick cell wall. Gram negative, inner membrane, super thin cell wall. So the big defining difference, other than that, is the fact that your gram negative is going to have an extra outer membrane. Let's color this in so we can... So, gram-positive, inner membrane, super thick cell wall. Gram-negative, inner membrane, thin cell wall, outer membrane. And this plays like a huge role in gram staining. So, first off with gram staining, you're going to use crystal violet, which is going to stain both cells regardless of the type purple. So you're going to use your, that's my marker is dying. You're going to use your purple at first, right? So both of them are going to stain purple. And then you're going to use Graham's iodine. Let's do this. Okay, so we add crystal violet first. Then we use our, which is our primary stain. Understanding like the names of these will also help because she'll use the terms interchangeably. So she'll be like, you first used crystal violet or you just added iodine. What is the name of that step? You would say mordant. If she asked, you just added crystal violet. What's the name of that step? Primary stain. So, we've got our primary. Okay, and then we've got our mordant, right? Which is going to be Graham's iodine. Okay, cool. Crystal violet, Graham's iodine. So the next step is the decolorizer, and I'll write that down here. Let's do the positive in here and the negative. So we've got our decolorizer. Okay, at this point, this is when you can start kind of seeing a little bit of a difference, which is something she might ask too, like when do you see the first difference within the Graham staining technique? You would say right at the decolorizer, because what's going to happen is this is going to remain purple, whereas this is all the color is going to be washed out. And for that, you're going to use alcohol or ethanol or hydrochloric acid. Actually, just say alcohol. Okay, you're going to use alcohol there. So, yeah, we only use alcohol in this one. So you're going to end up using alcohol here. And the reason why this happens is because see how this cell wall is super, super thick? Well, it holds on to that stain really, really well. So it ends up staining the whole thing purple. Whereas with this one, it only stained the outer membrane. So the outer membrane ends up getting dissolved by the alcohol. It'll poke holes in it, and then all of a sudden, you've got your cell wall, and you've got your inner membrane, but nothing is stained anymore. So then what you'll do is you'll add your counter stain. Okay? So you add your counter stain, which in this case is saffronin. Giving you the end result of what your Graham stain is. So here, it's still going to stay purple. And here, this one's going to turn it red. It honestly looks kind of more pink, but it's going to turn it red. So, to recap with your Graham staining technique. You add your primary stain, which is your crystal violet, which is going to stain both of them. Then you add your mordant. And we'll still draw that it's purple here. Okay. So then you'll add your mordant, which kind of just like seals in the stain, which will leave them both purple. Then you'll add your decolorizer. So your Gram positive is going to be purple, and your Gram negative is going to be clear. And again, to recap, the reason why is because this cell wall is so thick, it really can't dissolve the cell wall and like take all the color out. Whereas this one, it only stained the outer membrane, so it can poke holes within that outer membrane that will let you, and like dissolve the outer membrane essentially, which can let you stain this inner wall. And then you add your counterstain. Now, the counterstain really isn't going to have much effect on the gram-positive because it's already got that purple in there. And the purple will greatly overpower the pink, or the safranin. So the gram-positive will stay purple, and the gram-negative, since it had no color, since all the color had been washed out, will turn red from the safranin. Gram staining is so cool. I'm so excited to show you guys the pictures I took. Okay, up to this point, the big things I have gone over are biofilms, which just kind of help keep the bacteria alive and protect it from antimicrobial agents. Then we've got the people. I have a awesome slide that has everyone and their contributions to microbiology in that. So look at that, make flashcards of that. That's a really good one to make flashcards for. Then we've got recombinant DNA technology, where basically you've got your original DNA plasmid, and you've got some other gene, and they combine so that when that cell replicates, it has both of those DNAs in it. It's got whatever that new DNA is, plus the original plasmid. That's the big idea behind recombinant DNA technology. We've got taxonomy. Remember, it's domain, kingdom, phylum, class, order, family, genus, species. So, or specific epithet. If she asks specific epithet, species. And the way to remember that she told me years ago, 2 years ago, was kinky people cry out for good sex. Just remember to put domain first, before the, kingdom. And then we have 3 main domains, which are archaea, bacteria, and eukarya. And archaea and bacteria are prokaryotic, and eukarya is the only domain that's eukaryotic. Then we've got chemistry, where we talked about how atoms are basically assembled, right, with their protons, electrons, neutrons. And we talked about bonds. So covalent, where they're sharing electrons, ionic, where they're sharing, they have attractive charges, and hydrogen bonding, where hydrogens will bond with an oxygen or a nitrogen, and it's more of an attraction to their partial polarity that they have. And then we've got pH. That each going up the pH scale from 1 to 2 and 2 to 3 is 10 times more of a hydrogen concentration. That's the big note there. So if she asks any questions on that, I think she has a really good one on the practice exam about pea soup or something like that. It's just talking about finding that difference in hydrogen concentration between different numbers and different areas on the pH scale. Then we talked about anabolic processes. Remember I said to think of someone that's taken steroids, because it's called anabolic steroids, right? But they get super big and ripped and their muscles just get huge. They're building those muscles using the anabolic steroids. So an anabolic process builds something. An example of that is dehydration synthesis, where you take water out of the reaction from the original 2 molecules to make another molecule. On the flip side, you've got a catabolic process. And like I said, think of catastrophe, where things break, fall apart. It's a mess, right? So you're taking your big molecule and you're splitting it into 2 smaller molecules. An example of that is hydrolysis, where you're adding water to that original molecule to break it apart into 2 different molecules. Then we talked about macromolecules, which are our carbohydrates, our proteins, our lipids, and our nucleic acids. Remember that lipids are the only one that don't have a monomer. They're just kind of there. They don't have basic building blocks. Whereas you've got different building blocks for carbohydrates. Your building block would be a saccharide. And for proteins, your building block would be an amino acid. For nucleic acids, your building block would be a nucleotide. And then your polymers for all of those, you would have a polysaccharide, and you'd have a polypeptide chain. Polysaccharide for carbohydrates, polypeptide chain for proteins, and DNA or RNA for your nucleic acid. We briefly touched on protein synthesis, how you're going to have a DNA-like strand, and then it's going to be transcribed into an RNA strand, which will then be translated into a protein. And from there, we talked a little bit about like the different structures that proteins can have. Remember the example from my biology class that I told you about, where she'd go primary structure, and then secondary structure, or whatever, and then tertiary structure, where you curl up in a ball, and then quaternary structure, where everyone like congregates and curls up into a ball. I would remember that. Super important. And then ATP, just remember that the structure is most like a nucleotide, where it's going to have that nitrogen and like the nitrogenous base, which in the case of ATP is adenine, hence the A in ATP. But it's going to have that nitrogenous base. It's going to have a sugar, so a carbon ring, and then it's going to have a phosphate. The only difference between a nucleotide and ATP is the fact that ATP has 3 phosphates, whereas a nucleotide only has 1. And that was it for chapter 2, and then we got into chapter 3, where we talked about like the SI unit of the meter and how to convert from meters to, we briefly talked about how to convert from meters to millimeters, nanometers, micrometers, and just understanding the difference in between like the base units of like how small they are compared to it. Like millimeter is 10 to the third, I can't find a good way to say this. Is it 1,000? Oh, I really struggle with this stuff. Okay, I'm gonna write this a little bit just so, no, I'm not. Yes, I am. Let's slide down here a little bit. Okay, so you've got a meter, right? Then you've got your millimeter, which is essentially 10 to the negative third. You've got your, where did my paper go? Your micrometer, which is 10 to the negative sixth, and you've got your nanometer, which is 10 to the negative ninth. And that's just how small they are compared to that. So, like, 1 meter has, like, this much, if that makes sense. Sorry, I'm super bad with SI units, but if you can understand dimension analysis and understand that 1 meter is pretty big compared to your nanometer, like huge compared to your nanometer, like 10 to the ninth more than your nanometer, then you understand. It's just kind of like an inverse process. Then we talked about total magnification and how total magnification is just your objective lens multiplied by your ocular lens, and not to assume that your objective lens is a certain like power or that your ocular lens is a certain power, but to actually do the math behind it, because total magnification could say that your ocular lens is has a higher power than you originally thought. Then we talked about the FA technique, which is super cool. It just involves adding a fluorescent molecule to an antibody and then binding that antibody to a bacteria and then using microscopy to look at that, which is honestly, when you look into it, is super, super cool. Yeah, it'll light up the whole specimen so you can see it really easy. And she talked about that under the fluorescence microscopy slide, or right after the fluorescence microscopy slide. It's really, really cool. I think the specimen she used was a spiralium. We talked a bit on staining. So with a simple stain, you're only using that to look at morphology. And remember we talked about coccus versus bacilli versus spiralium. This is a super, super long recap, but coccus versus bacilli versus spiralium, and the difference between staph and strep. And then we talked about negative staining, which is staining the background but not the specimen. And then we talked about acid-fast staining, which is used with mycobacterium. And your mycobacterium should be stained pink, and everything else will be stained blue. And then we talked about gram staining, this lovely thing here, and just understanding the difference. So, like, with acid-fast, your primary stain would be your carbofuchsin, your decolorizer would be your alcohol, and your methylene blue would be your counter stain. Here we add that mordant of grams iodine to help hold in that that crystal violet. So, I know this was a really long review. Thank you for bearing with me. Feel free to email me if you have any questions. You guys are going to do great on this test. I believe in you. Flashcards, good ideas for it. All of the important people, maybe even the staining techniques if you're still a little unsteady on them, and definitely the microscopes, because I really want to help with these microscopes. I do. I'm very sympathetic to your cause, but I had a hard time with them, and I really can't help you memorize microscopy. Like, each of the different microscopes, that's something you have to memorize yourself. Because I can tell you the difference between a bright field, dark field, and phase contrast light microscope. But honestly, unless you know what they are, and even me telling you that isn't going to help you remember it any better. Because I'll just tell you what's on the slides. So, good luck. I believe in you guys. You'll do awesome. Have a great weekend.